My guest today is Dr. Yehezkel Ben-Ari. In 1971, Dr. Ben-Ari received a PhD with a focus on neuroplasticity and followed with postdocs at prestigious universities, including University of Cambridge, University of Oslo, and McGill University. In 1986, Ben-Ari was appointed head of INSERM in France, focusing on neurobiology and pathophysiology of development. In 1999, he founded and led INMED, Neurobiology Institute of the Mediterranean. In 2008, Ben-Ari turned to startups to better understand and study brain disorders. He is the founder and CEO of Neurocholre, where they trial Bumetanide and research the molecular mechanisms of autism, and BNA Therapeutics, where they study treatment options for Parkinson's and the regulation of intracellular chloride effects of GABA signals, and BNA Oncomedical, where they are dedicated to treat brain tumors. Dr. Ben-Ari is among the top 5 most quoted French neuroscientists and has over 520 publications and is a recipient of many awards, including INSERM Grand Prix, Grand Prix European Society of Epilepsy, Grand Prix Gagne Van Heck Belgian National Scientific Research, to name a few. Dr. Ben-Ari has over 5 decades of academic and private research with major discoveries on electrical signals of brain development and pathology, including a favorite of mine. GABA is excitatory during the developing immature brain. Recently, Ben-Ari is part of a team using machine learning to identify autism in the embryo and a pre-symptomatic signature called Neuroarchaeology that predicts brain disorders in utero within the developing brain, such as during cellular migration. Remember covering different epochs in the autism and embryo episode and how cells migrate to common cells to carry out common functions. Here, we will explore more on this critical time. And now, my conversation with Dr. Ben-Ari.
The issues I'm going to discuss pertains to the way one can identify early autism, how can one treat it, and why is it that studying brain development is crucial to understand and treat autism. Because the first thing one must remember is that autism is born in the womb, and this is what I'm going to show in a minute.
Okay.
And as it's born in the womb, it modifies developmental sequences which are crucial for brain construction. This implies that we're not going to cure autism in the sense I have a headache, I take an aspirin, I have no more headache. Because there are some small malformations in brain construction which are already present in utero. Meaning that we, at best, and this is not bad at all, we can attenuate strongly the disorder with a variety of drugs I'm going to talk about, particularly if they are started to be used very early in life. For a very simple reason is that the first years of postnatal life, once you are born, are crucial for many, many fundamental issues, social interactions. I mean, if your mother is crying and you laugh because you're looking at her shoes, it's not a lack of empathy, it's an incapacity to look at the eyes. So there are also very simple reasons why autism is what it is. And the first 2 years are absolutely crucial in addition to the maternity period. So first, let's go to the prediction of autism. Okay, this I remind you a couple of fundamental issues. The incidence of autism is going up. Nobody knows why. I think it's because of maternity problem, but this is my point of view. It's born in the womb. It can be due to genetic mutations, pesticide, pollution. No, go back to the previous slide. Yeah, it's generated in the womb by pesticide, pollution, a variety of factors. But the question is, if it's generated in the womb, can we identify the baby at birth who is going to be autistic and identify autistic years later? And the answer is yes. Next slide. What we did is the following. Well, this we can come back to this later. Go to the next slide. It's not the issue now. Okay, what we did is the following. We went to a maternity hospital in the south of France, and the pedopsychiatrist Eric Lemonnier identified 100 or 150 children with autism he was treating. He went back to the obstetrician, Dr. Kelly, which is here below, and this guy extracted the maternity files of those kids. In other words, you have a kid who is autistic, I am going to extract all the maternity files corresponding to that guy, from 3 to 15 or 18. In France, maternity files are very rich, between 120 to 150 different parameters, including at least 3 echographies, 3 brain scans during maternity, paid by the state. So, I mean, we have the collections. Taking all these parameters, I then recruited 2 big data analysts, shown here, INMED and Marilyn. They are experts of big data analysis, because you understand, if you want to analyze 120 parameters in thousands of children and different conditions, you need big data analysis. So what they did, now in addition, we took 3 times more kids born in the same maternity, in the same conditions, but not autistic, as a control. So you have autistic kids, non-autistic kids, born in the same maternity, the mother having the same age and same conditions. And then their task of these guys was to see if we can identify the babies without any a priori, the day of birth, not before. We need all the parameters going from conception to 2 days after birth. Otherwise, my trick doesn't work. And I'm stressing this because I've been accused of being for eugenics, which is, of course, I'm not. And killing a baby at 2 days old, it's not eugenics, it's murder. So I'm not in favor of it. So it's really after birth. The whole thing requires, it's very important because parturition and birth are crucial events in this scenery. So I need parturition and birth. Then they analyzed all this. Next, click on the same slide. Yeah. And what we discovered was we can identify almost 100% of the babies 2 days after birth who are not going to be autistic, they are neurotypical, and almost half those who will be autistic years later. Okay? Now, this is one trial. We hope to do more trials, more than 41%. We anticipate to be able to reach perhaps 60 or 70%, which, by the way, is higher than any identification of autism relying on genetics. Much higher.
Yeah.
So we're doing at least as good as genetics, in fact, much better. I'll come back to genetics later. It's my favorite topic, and you understand why.
Yeah.
Okay, next. So, then the question was, what are the parameters that brought the computer to decide this baby is going to be autistic? I mean, why has this decided this, the computer? It turned out that some of the parameters were expected. If the mother had rubeolis, cytomegalovirus, viral infection, etc., etc. Some were completely unexpected. But this I anticipated because I'm not trying to understand what happens. I'm simply trying to diagnose. Okay? Mm-hmm. One parameter interesting was, you see here in the middle, the position head down toward the vagina to get out for birth comes earlier in future autistic babies than in others. They are prepared to get out earlier. Now, a second important parameter, there are many other funny parameters. One of them is the size of the femur second trimester. I have no idea why the femur should be bigger in fetuses of future autistic. I have simply no idea. Yeah. But I don't care. I'm not asking that question.
Yeah.
It's too ambitious to explain that. Next slide. But then it becomes even more interesting because we discovered that when you look at the brain size in utero, second and third trimester. The brain size of future, we're talking here about fetuses or embryos, the brain size of these fetuses or embryos is bigger in 40% of the future autistic children already second trimester.
Mm-hmm.
Why is that important? Because a variety of American groups have shown that babies and infants have a bigger brain if they're autistic than non-autistic until adolescence. What I'm saying here is that this starts in utero, in fact. Brain construction is deviated by whatever insult led to autism. What's also more interesting is that if you look at the other side of the curve here, you can see that before delivery, the last image we did a couple of weeks before delivery, the brain of autistic children is significantly bigger than non-autistic children, all of them. As if in preparation to birth, the brain continues to grow. Because just before birth, it doesn't make any sense why they should grow more than neurotypic ones. But of course, and here I come to the issue of parturition. Parturition is crucial because it's by far the most complicated biological mechanism in mammals, by far the most complicated. There is no more complicated issue than parturition and birth. Among the fundamental issues in parturition is that the brain must be protected from the condition of delivery, which are tough. To give you one example, the Norwegian have shown that a baby born by vaginal delivery has a much bigger, has a different sensitivity to pain than if he is born from C-sections. They took a drop of blood and they measured how long he cries, basically. The idea is that oxytocin, and we show this in mice, during delivery plays an important role as a neuroprotective agent and a neuroanalgesic agent. Because there is pain, of course, when he gets out. And what they did in Norwegian was to measure the amount of stress molecules in just born babies. They show that the number of stress molecules of a baby born by vaginal delivery is huge, much bigger than the mother who is suffering, much bigger than you and I if we do 5 marathons or 5 hours sauna or God knows what. Now, this high concentration of stress molecules is crucial for the lungs, because the lungs, to evacuate water, need that. But the brain doesn't like that. The brain, if your brain or mine were subject to this concentration of stress molecule, would be epileptic. So we need something to protect the brain from this while keeping this for the lungs. My suggestion is that oxytocin does the job, because we showed that oxytocin in mice shortly during delivery reduces a lot intracellular concentration of chloride, reducing thereby ongoing activity. And of course, if brain activity is low during delivery, it will be less susceptible to anoxic insult, because there is a direct link. If you are hyperactive and you have an anoxic insult, your neurons are going to suffer much more. So oxytocin plays sort of role in addition to triggering delivery by provoking the contraction of the uterus. In addition to this, I've suggested, but of course, it's not proven in humans, it's only proven in mice. I cannot prove in humans, I cannot measure before and during contraction and whatever. It's very difficult to prove it in mice, but in human. But in mice, it is proven. So, what we showed in mice, which is quite amazing, next slide, I think, if you click again, this is the same one. So, future infants, no, let's go back. Go back, one more. Yeah, here, stop here. Future infants with ASD have a bigger brain than neurotypic shortly before birth. Now, what we showed in mice, not in humans, is that if we measure the size of neurons in the cortex 1 hour before delivery and 1 hour after, but taking different mice, sacrificing them and recording neurons, the neurons continue to grow in autism and not in controls. It's as if in naive animals, the brain stops growing, which makes sense. Not in autistic future mice. It goes on growing during this very tough period called parturition and birth. This we cannot prove in humans for obvious reasons, but we have proven this in mice. Whether it's true in humans is open to debate, but it's not unlikely that this would be the case. So, brain growth is impacted in whatever mechanism leading to autism in utero, leading to bigger brains of fetuses and embryos, and leading to something which is reminiscent of a different preparation to delivery and birthhood. Okay? Next. This is the conclusion of that part.
Okay.
Now, the advantage of making such an early, by the way, the paper we published was the first worldwide demonstration that you can identify babies at birth will be autistic, and there is no genetics here. There is not a single genetic parameter. Forget about genetics. It's useless.
Yeah. It seems like it's a waste of time. It's a dead end.
Time and money. Unfortunately, it dominates for reasons which are very mysterious.
Yes.
I mean, I had discussion with Simon's Foundation on this. I failed to convince them how genetics is a complete waste of time and money. But this is
I'm glad you put that out.
Because the facts are stubborn. It's 4 decades promising the moon and nothing happens. There is not a single treatment based on genetic information.
Yeah.
Come on, okay. And I am providing an explanation here of that is the case. Okay, next one. Now, how would I treat autism? Next one. Now, this slide summarizes what I have called the neuroarchaeology concept. The idea is the following. Usually, people interested in genetics and all these models of proteomics, genomic, metabolomic, and God knows what, they want to treat the inaugurating insult. So you have an insult associated with deficit A. I'm going to compensate the deficit A to block the inaugurating insult. Now, when the inaugurating insult takes place in the womb, there's no way you can do that by definition. Why? Because autism is a clinical diagnostic disorder. It's not a biological defined. I mean, a child is considered autistic when the pedopsychiatrist says he's autistic. There's no biological signature, straightforward one. Now, if the insult comes in utero, forget about gene therapy or treating the inaugurating insult. Why? Because what I'm going to show now is that the insult modifies brain construction, and the disorder, when you diagnose it, is due to the modification, not to the initial insult. The initial insult is forget it. You cannot correct it, it's too late. For instance, if the inaugurating insult has led to a bigger brain or to misplaced neuron because they haven't migrated, there's no way you can correct this by genes or anything else. Why? Because there is no more migration. The space is taken. I mean, there is no mechanism allowing migration to take place. So the neuron which is misplaced will remain misplaced.
Yeah, its final result. Its final.
So you cannot. So what you must do is not treat the initial insult, but the consequences of initial insult. If you click again, this arrow is going to move to the right. You want to treat the target, not the initial cause. It's completely useless. This is one of the reasons why genetic stuff is boring and totally inefficient. Next one. Now, this is an important slide because it summarizes something which is simple and complicated. The basic idea is what? The basic idea is that I discovered 40 years ago that neurons in the mouse brain have high concentration of chloride when they are immature and low when they are adult. Now, they have high concentration of chloride initially in development. This has been confirmed in every animal species until human, all the way to humans, from fish to birds to whatever. It has been preserved throughout evolution. In all animal species, chloride is high initially and low subsequently. Now, this is important. Why? Because the principal inhibitory transmitter, GABA, which provides brain inhibition in adult, in young neurons excites, doesn't inhibit. Why? Because its actions are dependent on the level of chloride inside. If you have low level of chloride, it inhibits. If you have high level of chloride, chloride is going to exit the cell instead of entering, so the polarity is reversed. This means, for instance, that a pregnant woman taking Valium, which acts on GABA, she might have different effect on her brain than that of the fetus, because the system is opposite. To get a long story short, the developing brain is not a small adult brain. It has its own rules, its own mechanism, its own function that are not present subsequently. You must look at it as a special beast, so to speak, not common with what happens later.
Yeah.
This is quite important because it means that drugs, for instance, all sorts of common drugs, Prozac and God knows what, should not be taken by pregnant women because what they do on the mother is not necessarily what they're going to do on the little one.
Yeah, and that's becoming more and more understood and accepted.
Yeah, but I mean, basically the idea is that during pregnancy, women must abstain of almost everything. Unfortunately, this is the way it goes. Pollution, etc., etc. For everything, there is evidence. Everything: pollution, stress, molecules, whatever. Now, I discovered this. Then I discovered that in autism, there is a reverse situation. In cortical neurons of mice with autism, GABA again excites because chloride again accumulates. Now, the interesting thing is that in both cases, it's a reverse image because in the developing case, chloride is high because there is a co-transporter called NKCC1, sodium, potassium, chloride, NKCC1, which is crucial. It's present in all cells and it controls ion distribution. As you know, neurons are like a battery. All their energy is spent to preserve ion distribution. Low chloride inside, high sodium, high potassium inside, low sodium outside, etc. If this is deviated, you are epileptic and you have all sorts of disorders. Now, in pathological conditions, again, NKCC1 is overactive, as if the neurons are again immature. This is an oversimplification, but that's the idea. This was reported in autism, in epilepsies. Then it was discovered in all the disorders I'm listing here. It's an incredible list of disorders in which some neurons are again immature, basically. Now, this means that drugs blocking NKCC1 or activating, you see, the chloride concentration is controlled by an importer and an exporter. The importer is NKCC1, the exporter is KCC2. And in pathological conditions, the exporter doesn't work well and the importer works a lot. So chloride accumulates and GABA excites. Now, in all those conditions, it works, meaning that a drug blocking NKCC1 is going to be very useful in a lot of disorders. So we investigated two pathological conditions. One I'm not going to talk about, I'm working a lot on this, and this is brain tumors, but I want, there's not a subject of your, I'm going to talk only about autism. Next slide. So what we did, basically this situation, you have low chloride normally, high chloride in pathology, and if you block NKCC1 with this drug called Bumetanide that's shown here, this is a generic drug fabricated 50 years ago by a Swiss company to treat hypertension and brain edema. Basically, you go to the loo, PP, you remove water, and your tension is reduced, etc. Now, in theory, Bumetanide should be able to block all of these. Now, this is not only a theory, because this has been shown to be the case experimentally in all these disorders, animal models. Next one. Now, the NKCC1 that you can see here is crucial in a lot of systems. It consumes a lot of energy via sodium, potassium, ATPase, and ATP. Its structure has been crystallized more or less. You can see to the right. And Bumetanide, we know more or less at which site it penetrates to do what it does. And Bumetanide is the best and most specific antagonist of NKCC1. In fact, NKCC1 was partly cloned because of Bumetanide, thanks to Bumetanide. Next slide. We did 2 phase 2 double-blind randomized studies in France. 84 children, and I think 6 or 7 children. In both cases, it was bumetanide placebo, children 2 to 18 or 3 to 11, treated for 3 months with half 1 or 2 milligram bumetanide. I can show you films. In several cases, the effects are spectacular. Now, if you see this here, why do you see this? What you see at your right, every blue column here is one kid in this trial. The measure is called CARS, Children Autistic Scale. This is done by the pedopsychiatrist. The bigger the column, the better the effect. If you have 8 to 10 points amelioration and you go from 46 to 36, you're not cured, but you're very strongly less autistic. And you can see that all the blue columns are treated, with only one exception, orange here, which is placebo. So there are exceptions, of course, but according to this, the treatment is quite efficient. Then we did it with, at the right, the table that you have is social responsive scale. This is another scale made by the parents this time, okay? Here we gave 0.5, 1 or 2 milligrams to the kids. According to the FDA, if you have more than 10-point ameliorations, it's extremely good. You can see here below the last line that we had between 10 and 20-point amelioration and only 1.5 point amelioration with placebo. So the difference is quite spectacular.
Yeah.
Of course, we abolished 2 milligrams because the diuretic effects are too strong in a young child. So we decided to take only 0.5 or 1 milligram twice daily, morning and afternoon. And this is now used all over the world. I mean, next slide.
More eye contact even.
A meta-analysis, in fact, revealed that 1,050 children have been treated successfully in 9 trials, our 2 trials, and 7 others made in China, England, Holland, Egypt, Tunisia, Iran, and I forgot which other country. All of them doing exactly what we did. 0.5 milligram twice daily, 1 milligram twice daily, 3 months. All of them showed a very significant amelioration. The Egyptian stuff is quite interesting because they isolated the parameters, which seem to be particularly improved, and this is agitation and social recognition and frightened of novelty, facing novelty problems. So we fabricated a liquid formulation of bumetanide because the pills are very non-tasty and big. Next slide. These are the trials made in different countries confirming what I just told you.
Okay.
Next one. Then we did visual interactions and brain imaging on 15 adolescents with autism before after treatment. What we found is that after treatment, the kids recognize better the emotive figures that you see here, which are different on the neutral ones shown at the right. So we asked them to recognize the woman if she's crying or laughing, whatever, and the guitar or the rabbit or whatever. And there was significant improvement. Next slide. Then we did brain imaging. One more. And we found that regions, if you ask the child or the adolescent, these were the adolescents, to look at the eyes by putting a cross on the nose and asking the kid to look at the eyes, usually they're frightened. The activation by the image of the eyes of the amygdala, which is the fear region, is reduced by bumetanide. They are less frightened. And we have a lot of direct proof of this, that they look more at the eyes after treatment. Next slide.
Can I ask a question real fast?
Sure.
Is this interacting at all with oxytocin or vasopressin?
Now, the problem with oxytocin, vasopressin, to the best of my knowledge, there is not a significant demonstration that it's efficient in humans in clinical trials. To the best of my knowledge, there isn't. Oxytocin, there have been many, many trials in Europe, in the US, and in Japan. Unfortunately, they are very, very contradictory.
Yeah,
Now, remember, oxytocin does exactly what bumetanide does. Reduce chloride and inhibition. I agree with that. The problem is the lifespan of the drug, how fast it's degraded, and so forth. In addition, and this I'm going to show in a minute, you're going to understand why oxytocin results have failed. Just keep the question for one more minute. Next slide.
Okay.
All right. Now, unfortunately, in spite of all these results, we did a phase 3, final phase 3, on 420 children in two groups, 2, 6, 7, 18 years old, in Europe, Brazil, Australia, 40 centers, a very large study. It failed. There was no difference between treatment and placebo. Now, phase 3, the problem is you have only one parameter that must differ in placebo and treatment. And this parameter, for instance, CARS, is a mean of 15 different items. SRS is a mean of 65 different items. Meaning that if the mean is not different, it does not imply that sub-items coding for agitation, visual connections, seizures, whatever, will not be significant. You see the point? Centering everything on a mean of something so heterogeneous is to some extent meaningless. This, by the way, explains why all phase 3 trials done on Fragile X, Autism X have failed, even though phase 2 were successful. Phase 3 fail always. Heterogeneity and lack of taking into account the multiplicity of items that can be modified separately. If I have a drug that treats agitation in kids with autism, it's not bad, even if it doesn't treat something else. You see what I mean? Yeah. So what we did, again, we did the same thing. And this was also influenced by a study made by a Dutch group showing that you can identify by EEG the kids who are going to respond to bumetanide. The Chinese showed the same by measuring immunological sample, amount of stress molecules. The more you have stress molecules, the more you are going to respond to bumetanide, basically. I took a very different approach. Next slide. Again, going back to machine learning, we decided that all this data we have a huge amount of data on phase 3, hundreds or thousands of parameters. What about using machine learning to identify subpopulations? So what the computer does, it takes all the kids with all the parameters, not only cars, but all the 15 parameters, sub-items of car, 65 sub-items of SRS, etc., etc. And he identifies subpopulations. Okay? The question was, can I identify subpopulation of kids responding to bumetanide within the phase 3? And can I identify them in order to be able to do a subsequent phase 3 in which inclusion criteria will be the ones I have defined? So I would not include all children with autism. I'll include children with autism having those features. And we succeeded. We have identified 25 to 40% of children responding to bumetanide, relying on clinical sub-items, a mixture of several sub-items. This is important. Now, this is now being, I hope, patent protected, because this is a private company. I don't have a huge amount of money. I need money to go on. I don't have any financial support for the moment. And I'm trying to convince a pharma or an investor to help me making a phase 3 with these criteria. This will succeed.
Everybody's money is going to genetics, though, as we discussed. So,
no, yeah, but I mean, you see, they have a sense of humor. There are experts in genetics who have published every month a paper in Nature on the new mutation, blah, 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 blah. They have never done a clinical trial. You know what they're doing now? They want to use lithium to treat autistic children. Give me a break.
Yeah, and I'm seeing more antipsychotic treatment, too. And
do you need to make thousands of studies on specific mutations then to use lithium? Give me a break.
Yeah.
It won't work, by the way. It won't work, we know.
It's so bad.
No, but they are desperate to propose a treatment. Desperate. Because using stupid, old-fashioned drugs, reach phase 3. What do they have to say in front of this? Nothing.
And it's just an antidiuretic, correct?
It is a diuretic. It is diuretic.
Yeah.
And the only side effect, in fact, indeed, are diuresis and dehydration. But this has been given to millions of patients during the last 20 or 40 years. There's not a single death. We never found anything really serious. Nothing. Yeah, I agree. You must control potassium down there in the blood, and that's it. And drink water and go to the loo. It's all right. I mean, the side effect of some of the drugs used for epilepsies, or for autism, by the way, like the two favorite ones, people get fat. Forget about this. No, there is no comparison with other drugs. So, but the message here is, not only it's not genetics, it's really maternity. And it's really maternity with, by the way, the Americans in San Francisco have proven that a woman pregnant second trimester close to a field in which you have pesticide, the incidence of autism goes up. It's proven. It's not stupid ideas of genetic guys. It's the same thing with pollution, the same thing with stress, the same diabetes, preterm delivery, C-section in certain cases, etc., etc. All these relate to the womb, maternity, and delivery, meaning that you really must protect pregnant women. This is the most important message of this. And you know, if they have diabetes, they are poor and they eat fat, and etc., etc. You know, the consequences are well known. The problem with genetic, it is too deterministic, very stupidly, because life is not deterministic. I mean, you know, you and I have the same genes. If you go to eat every day to McDonald's, I don't. You eat diabetes, I won't. It's as simple as that.
And plus, like chimps, we're pretty much identical genetics with chimps, and we're nothing like chimps.
No, okay. But by the way, they rely a lot on twin studies to decide its genetics. I'm just writing a paper, I hope to publish it soon, in which I'm demolishing point by point everything about genetics. I'm explaining that twin studies are wrong. I'm explaining that what they love most is called GWAS. I don't know if you know what GWAS is.
Yeah.
GWAS is polygenic, whatever, blah, blah, blah. I mean, the basic conditions to use GWAS are never met. Never. It's all blah, blah, blah. And I'm writing a very detailed paper. Now, of course, I sent it to genetic journals. They rejected it. But now I sent to a medical genetic journal, perhaps it'll be published. It's very, and I'm writing a book in English with a colleague of mine, with expert evolution. The title is something like Autism is an Neuroinflammatory Disorder. Forget about genetics. Well, anyway.
Good.
Let's go back to our topics. Next one.
Okay.
Okay, so this is summarize what I told you.
Okay.
So we're aiming to find money to use a phase 2-3 on identified responders, which will work, bringing the first drug within 2 or 3 years to the market to treat autistic children. Next one. However, in addition, I did something else. I decided, you know, pharmas don't like generic drugs because you can buy it in the pharma, it's cheap, and they're not going to make money, basically, to cut a long story short. So I decided to fabricate molecules which belong to me, novel molecules, which block NKCC1, belong to me, and are as efficient as bumetanide, and perhaps even better. Next slide. So working with chemists for years, 3 years now, we are looking at this. Well, forget about glioblastoma, it has nothing to do here. But anyhow, we wanted drugs which work as well as bumetanide, and our aims are to find money to go to pre-IND studies to phase 1. Basically, if you have a new molecule, in vitro it does what you want. We show that it does exactly what bumetanide does on seizures, on activity, on GABA, etc. Well, now we need to take the best one to pre-IND phase 1. And this costs a lot of money because toxicology, genotoxicity, etc. But, next slide. We have now 120 different molecules. They all act on NKCC1. Some of them are better inhibitors than bumetanide, which is, I don't think anybody has discovered a molecule better than bumetanide. I've tested them in cancer. Sorry, this year there is a mixture with cancer. But forget about cancer. The idea is perfectly the same for autism. Now, the idea is that if I have a molecule reaching phase 1, not toxic, it's going to be efficient in so many different molecules that it's going to be extremely valuable. I mean, I guess even billions. Because it could work for us. By the way, there is a beautiful paper on Alzheimer's disease, which I liked a lot. 2 years ago by an American group. The first author is Taubes, Taubes, Nature Aging. What they showed was they tested 1,000 molecules, which are widely used, Valium, phenobarbital, Prozac, whatever. Which of them is the best on ApoE-dependent Alzheimer's disease? The answer is Bumetanide, out of 1,000. Then they did something which is even more impressive, epidemiological study on 6 million 65 years old Americans, comparing those who took or not bumetanide for any reason. Those who took bumetanide have a reduced 30 to 70% incidence of Alzheimer's disease. Which I think is very, very striking. So this drug is not dead. What I'm trying to say, the phase 3 died, not the drug. Next one. I think it's the last one.
Okay.
No, this is simply physiology to show it does exactly what bumetanide does. In rodent brain slices, it does exactly what you expect, blocking seizures and reducing GABA, increasing GABA inhibition. I think that's it.
Yep.
Have I summarized everything, or are there other questions?
So I would like your idea of the neuroarchaeology and identifying within the womb, and you comfortably saying, I think pretty confidently, I should say, that autism is definitely in the womb. What do you think is the biggest barrier for getting that out as a being accepted, both by other scientists and how to allow the lay public, just normal common people, to understand this and understand the risk of the environment for the pregnant mother?
This is a very fundamental question. Now, the answer is complicated. A, because it's also to some extent political. In countries like France, pregnant women are protected. They have 12 weeks vacation, all paid by the employers. To take only one example. In the US, it's not the case. So there are political aspects I will leave aside. But the fundamental issue is very simple. The data we published, and many others, by the way, following our results and others, are compelling. I mean, the only issue with the public, which one should be very careful about, is the problem of it's the mother problem. She's responsible, which has been here for decades. It's a very stupid argument, which makes no sense at all. It's not the mother responsible. If the mother lives in a field in which you have pesticide, okay, the pesticides are going to do what they do. If her her husband is alcoholic and beats her, and God knows what, and she has nothing to eat, or God knows, is she responsible? I mean, the responsibility of the mother is something disgusting, to be honest. It makes no sense.
I agree.
I'm saying here that during maternity, this is born for reasons that are so multiple that, but we're beginning to win. Women pregnant now smoke less than in the past. They take less alcohol than in the past. So we're improving, but it's very slow. And it's very slow because here I'm not talking about alcohol and smoking. I'm talking about life conditions.
Yeah.
You should try not be obese or diabetic when you're pregnant. There are hundreds of problems. Anti-epileptic is a problem. The woman is epileptic. There is an anti-epileptic drug treating very well as seizures. She becomes pregnant. Stopping the treatment is problematic because she could lose a child, the baby. Giving it up continues is also problematic because it could be malformed. And there are trials in France against Sanofi because a very classical anti-epileptic called Depakine has been shown 30 years ago to increase brain abnormalities and others abnormalities. But the decision is not simple. If I am the neurologist, I have a woman epileptic, what do I do? It's not simple questions. You don't want to stop it, and you don't want to go in either. So The message is any politics should do all its best to protect women during pregnancy.
Yeah. Yeah.
And this is it's not the priority today.
No. And as you mentioned, like it's much different in the US. The environment of the pregnant mother is typically not great. Um, and it might sound sexist or something like that, but they're forced to, or they choose to work up into almost the time of labor, and it's just very concerning to me.
Well, in France, you have 12, now 14 months even. You have protection for a long time, and the husband can take part of it to help whatever. By the way, I wanted to do this study I showed here on in utero measures to detect. I wanted to do this in the US. Spoke to some of my colleagues at the big hospitals in the US. It's impossible because the echography, the whatever, are paid by the patients. So you have very little information.
Yeah.
There is no real collection of maternity data. So I can't do it in the US.
Yeah.
So this stuff will be in France, not in the US. By the way, I cannot apply it as if immediately to England, because in England you have only 2 echographies, not 4, not 3 like in France. And they have less collection of it's depends on the countries. It could work in Norwegian country, Norway, Sweden, Holland, as usual. These are the best in terms of care of women, etc. Perhaps Switzerland. Yes, Switzerland probably. But that's it. Even in Europe, it would not work in Bulgaria or Romania or perhaps even Germany. So it's an endless problem. It's a very complicated one, yet a crucial one.
Yeah. There's a lot of humans that don't understand the problem involved in the decision making. Do you know the differences between the rates of autism in the US and, let's say, France?
I think they're quite similar.
Okay.
But the answers are not straightforward because in your country you have better epidemiologists doing this than in France.
Okay.
I don't think we have as detailed epidemiology on
There's
autism.
a lot of variables within that question.
Yeah, but I mean, we don't have the same amount of studies on epidemiology of autism in France.
Yeah.
But I guess the numbers will not differ tremendously.
And even across US, there are areas that have extreme rates, like around San Francisco. They have almost like 1 in 20 children.
Yeah.
So
No, it's frightening. But it's not only autism. It's a fact that if you make a very simple financial calculation, which your government and mine like a lot, it costs much more to treat them afterwards than to do what they can do on pregnancy. There's no comparison.
yes, I have an episode saying that. I'm glad you confirmed that.
Oh, the cost, I mean, people have calculated, I can find the papers, how much a child autistic costs to the society and parents. If you calculate everything, the woman not working, blah, blah, everything, and you include all these, the sums are huge,
Yeah,
billions.
I have an episode, autism.
You know,
It's
there is
just
no secret.
blasphemy.
In medicine, what's important is prevention, not treatment, always.
And
Always.
US, I can't speak of other countries, but the US is awful. They're probably the worst at this.
They are among the worst.
Yeah.
But the life expectancy of Americans is below that of Cubans.
Yeah, and it's very stagnant and flattened towards the bottom. Yet our health budget is almost 2 times the second.
You pay more than us and you have less life expectancy for a very simple reason. The reason is that socializing health is cheaper than non-socializing it. In our country, we have one social security. I paid my part directly from my salary. They take a percent of my salary to put in social security. Then I don't have to pay medicaments, treatments, surgery, whatever. And it's cheaper. No, it's again, it's a political problem. And, we're not going to go to politics, but this is really a political problem. And you're facing an election, which, well, I won't say anything, but
Yeah.
it become even worse.
Yeah. And the 4 of the top 5 revenue industry types are around healthcare, hospitals, big pharma, health insurance. But I didn't bring you on to talk about that, but it is an important topic.
I agree, but this is beyond our competence, yours and mine.
So whenever I first heard GABA was excitatory through that immature developing brain, I was blown away, because one of the first things I learned about autism was this excitation inhibition imbalance. So, and I assumed that GABA was excitatory up and through this probably critical period. Is it because it helps accelerate learning and development?
No, GABA is excitatory only in utero in humans, not afterwards. But it's excitatory in utero because we and many others have shown that GABA exerts trophic actions. GABA controls neural growth, synapse formation, network formation. If you block GABA, neurons do not establish as many synapses and do not grow as much. And migration is deficient. So GABA excites immature neurons for these reasons.
Oh,
And then when there is a lot of excitation by glutamate, it becomes inhibitory. Nature is much cleverer than we are.
yeah. Much closer. Wow. Okay. I'm looking forward to your future research, and especially some of those things that you're working on. As you get more results, maybe you can come back on and discuss those with me. I'm going to be looking out for some of those items that you mentioned today. I'm very
The thing is, hoping this paper will get out. The book will take some time.
yeah.
But I have 2 books you can advertise in English. One of them is The First 1,000 Days, which is published in English now.
It's a great video on YouTube about that.
And the second one is Bumetanide to Treat Autism. The 2 books are now in English, and they are accessible via the usual sources.
Okay.
In the first one, I explained why bumetanide treat autism is efficient. And in the second one, I explained why the 1,000 days going from conception to second years of life are crucial.
Yeah.
I'm going through everything I told you here. Now, in utero, womb, pollution, etc., etc. So the 2 books are related to what we discussed here.
Yeah. I get frustrated with autism research a lot, but you're one that I have trust in. You expand the edges of science instead of just being stuck.
You know, I have always said to my students, if you want to make great discoveries, they're always in sideways, never in highways. If everybody does this, you go this.
Yes.
Never followed modes and fashion. This is why I don't like genetics, because it's a mode and a fashion. It's going to die one day or another when they discover that. And the same thing applies to all these big stuff, proteomics, metabolomics, genomics. We're going to count every protein you have in your body, and we're going to understand why you or somebody else is autistic. Give me a break. If you take a transistor from a television, it doesn't work. It's a transistor, which in fact it doesn't tell you anything about what else happened. Come on.
I would like to give a special thank you to our guest today, Dr. Ben-Ari. It's not difficult to see Dr. Ben-Ari's passion towards autism, discovering the precise where and when autism is developing. His research is expanding the edges of the autism community. It's unfortunate that there are other factors involved with autism research, but we very much appreciate his voice for the autistic community, autistic research, and what is wrong. Now, we went into genetics, and it's not uncommon that the podcast speaks about the genetic factors and how there's not an answer here. You even heard Dr. Catherine Lord mention that she doesn't think there's an answer here. Yet, so many people are invested and they want an outcome here. They keep searching, but it's incorrect. I very much appreciate Dr. Ben-Ari's 5 decades of research and knowledge and information that he has contributed to both the scientific community at large and the autistic community. he has a lot of good projects going and moving forward. So this gives us a lot of hope and something to look forward to as he is publishing and finding other answers and other alternatives. I very much appreciate him speaking out on the psychopharmacology that's being used for autism. This is very concerning. If you're listening to the podcast or listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586, or you can check out the Hop link where you can find all links to the podcast forms and my contact information and social media. And thank you for listening to From the Spectrum podcast.